Hi, I'm Anna Dhody, curator of the Mütter Museum and director of the Mütter Research Institute. And today I'll be talking about the Mütter Research Institute looking to the past to improve our future. I always like to give the goals of my lecture so you can see when I'm not achieving them, but I definitely try. So I'm going to give a brief overview of the College of Physicians of Philadelphia, the Mütter Museum, and a little bit about our collection so you have a frame of reference. We're then going to discuss the creation of the Mütter Research Institute. Why did we do it? How did we do it? And then we're going to talk about a few of our ongoing projects, things that we are currently working on that involve our wonderfully, disturbingly informative collection. And of course, we always like to try to slightly disturb you. That's one of our goals. Now, first, I have to talk a little bit about the College of Physicians of Philadelphia. Without the college, there would be no Mütter Museum. So we always like to give a little bit of reference about that. Here in the United States, we often get very confused people who think we're a medical school. And of course, we're not. You know, we are a professional society of mainly physicians, about 1,500 or so, mostly located in Philadelphia, although we do have physicians abroad. And we are the oldest professional medical institution in continuous operation in the United States. We started in 1787, which sounds very, very old to us, but maybe not so much to other people. Now, the College of Physicians, like I said, was a professional society, and we started in 1787, like I said, but the Mütter Museum did not come around until after the 1860s, and the reason it's called the Mütter Museum is because of this handsome gentleman right here, Dr. Thomas Dent Mütter. Now, you'll notice there's an umlaut over the U, so that is why we are not the Mutter Museum, but the Mütter Museum. I've been told that I do not pronounce that correctly if it were the German word, but we do our best. So unfortunately, he was one of our fellows, he found himself in ill health. This was around 1858. And previously, he had been a very, very popular professor of medicine at what is now Thomas Jefferson University in Philadelphia. So he unfortunately had to retire, and he decided to bequeath his entire teaching collection to us. And that's very important because he had amassed this collection over decades of teaching, about 1,700 specimens, objects, things like that. And that's why it's called the Mütter Museum. He also did give us a substantial endowment of about $30,000, which in today's currency is about $750,000. He did have one stipulation, which was if we were to take his collection, we had to build a fireproof building to house it. Very smart, because of course a lot of alcohol-based wet specimens, so fireproof building is always a good idea. Unfortunately, he died in 1859, and we were unable to complete the building until around 1863. So the Mütter Museum has been open since 1863, and the College of Physicians since 1787. Those are our important dates. Now, here is an overview picture of the main gallery of the Mütter Museum, and you'll see that it is in keeping with what is considered to be the European salon style or cabinet format. There's also the mezzanine. I've been to many medical museums around the world, and the mezzanine seems to be a very consistent architectural detail that I see in many of them. The Mütter Museum is grouped in this main category, mostly along the lines of content, anatomical body system, things like that. And we've been in this kind of cabinetry style situation since we opened. In fact, these cabinets are original to the first building that we had, the one that opened in 1863. That was at a different location than where we are now. But the cabinets, at least some of them, were brought over to this new location. The building that we're currently in, we've inhabited since about 1910. And so we've been there ever since. It was built for us, too. So very briefly, I want to go over some of the types of specimens and objects we have in the collection. And this is important to kind of give you an overview of the breadth of the different types of things that we have. Mindful that the intent and the use for all of these collections has always been education. So that's very important. So wet specimens, I think these are the ones that we are most known for. That's the thing that people kind of associate with us. One of the most things that they do. And we have over 1,300 of these wet specimens. And they go by many terms. I believe in the UK, they're referred to as potted specimens, or we also can call them fluid preserved specimens. And again, the wet specimen, it's just any biological object that's preserved using a liquid fixative. Now, our wet specimens go back to the early 1800s in terms of a provenance, and of course that predates the creation of formalin or formaldehyde. And what's very interesting is some of our specimens were sealed up in their containers prior to the creation of formaldehyde, and have never been opened since. Now, some of our specimens come to us in formaldehyde, and then we have to exchange the fluid because we do not put anything on display that's in formaldehyde or formalin because that, of course, is carcinogenic, it's a caustic, and so all of our specimens are in a 70 to 30 alcohol water preserving solution. Osteological specimens, human bone specimens, of course, we have many thousands of those as well, and they're in a variety of different presentations. We have fully articulated specimens, we have non-articulated specimens, loose bones, things like that. Dried specimens, very, very self-explanatory. They have been desiccated, they've been dried out. This is our megacolon. It is, to my knowledge, and I always gotta give that a little caveat, the largest human colon on display in the world. It's 8 feet 4 inches long. It's about 1.5 feet in diameter. At the time of death, it held approximately 40 pounds of fecal material. That being said, if anybody has a larger one on display, please let me know and I will adjust my talk accordingly. Models, very important, very important. We always like to stress that our anatomical models are very, very important when it comes to education, the teaching of medicine, and we have a lot of very different specific types of models. Again, each one was used for a different purpose. Now, sometimes we have models that were used to capture a specimen, for instance, that we were unable to keep. A good example would be to my right at the top, that is the death cast of Chang and Ang Bunker. They were very famous conjoined twins. Upon their death, their wives gave us, the college, permission to conduct an autopsy, but we were not able to keep their bodies afterwards. So they did this cast to kind of get their form for educational purposes. At the bottom there, you'll see one of our wax models showing skin pathology, dermatological conditions. Again, very, very important. We've actually had some negative reviews on our Yelp sites and things like that, saying we've got too many fakes, not real wet specimens. And, we take exception to that in the sense that these wax models are extraordinarily important. They are over 150 years old, and they were designed for a very specific reason. Wet specimens, don't get me wrong, I love the wet specimens. They're very, very important. However, when you put a biological specimen in a liquid preserving solution, in most cases, that specimen, especially if it's something that involves skin, will lose its original color, and you won't be able to get a really accurate idea of the texture. And that's very important when you're working with dermatological conditions. You need to have an accurate representation of color, of texture, in order to learn more about it and to hopefully have a correct diagnosis if you're a medical student to learn about that. Now, the model that is in the center of the hand, that is actually about 5 to 7 times larger than the size of a normal adult human hand. This was used in the situations where you have students, medical students, in a kind of classroom with the stacked seating, the kind of stadium seating, and you have the unfortunate students who are at the very top would not be able to see very well. Of course, this is before PowerPoints, before overhead projection in most cases. And so if the professor was going to be doing a dissection on a average, human hand, people up top would not be able to see anything. So these larger models, these often paper mache, they're called clastic models. They come apart, and so they could be like virtually dissected, and you could see the anatomical structures underneath. Very important. Again, all harkens back to teaching, and that is the mission of the Mütter Museum, is to teach. And I think it's very important to understand the different ways in which we do that. Of course, we have instruments, chemicals, and drugs, all sorts of different things like that as well. Most of them have been donated by our physician fellows at some point or the other. And I have to, one of my favorites, of course, is this bottle of pills that's in the center here. First of all, I adore the color. That's one of my favorite colors. And hopefully you can see the contents of this particular bottle. It has digitalis, strychnine, nitroglycerin, and of course, cactus, because cactus. Just love looking at that. And of course, we just have so many other things that might not necessarily fit into the specific topics, like our soap lady. She's a one-off. She's a one saponified human body. We have large objects, of course, too, like our iron lung. We have an amazing collection of historical medical photographs. And then we're also still collecting, we'll go into that a little bit, but this image down here of this skull, it's actually created using over 100 acrylic preserved brain slices. So they are, they're not dried. They're not wet specimens per se, in the sense that they're not in a liquid preserving solution, but they are still wet-ish, I guess, and then they're encased in this plastic-based acrylic perspex type of material. So they're shelf-stable. They don't need to be refrigerated. They're very unique. So we had to actually, when we acquired them, had to create in our database a separate category to describe them, which is always new, always interesting. And like I mentioned, some are old and some are new. What do I mean by that? Well, obviously, we have some specimens that are well over 150 years old. The main bulk of our collection, I would say, is mid-19th century, because that was really the heyday of our particular collecting time period. So Dr. Mütter, of course, and our physicians, they really did the bulk of the donation of our core collection. I would say, again, 1863 till maybe the end of the 1800s, early 20th century. Of course, some of the objects themselves, of course, are very old. So to the left, you see an Assyrian medical tablet. This is a prescription tablet, kind of detailing the types of ingredients that you would need to relieve a certain condition. I believe one of those ingredients is beer. We have specimens, donations that come to us that are new for us, but they are older specimens. A good example would be up in the top middle. We have brain slides from Albert Einstein. And of course, he passed away in about 1955. So these slides date to about that time period, but they did not come into our collection until 2011. And we're actually, we are still actively collecting. And I know some medical museums are still collecting, not a lot to my knowledge, are actively collecting, but we are. And one of the things as curator that I'm very interested in collecting is representations of public health issues that are 20th and 21st century public health concerns, things like that. Our collection is wonderfully represented in the public health issues of the mid-19th century. So we have, a lot of syphilis, a lot of tuberculosis, infectious diseases, things like that. I'm trying to actively collect representations of the same public health issues, but that are very prevalent in the 20th and 21st centuries. So a good, and like I mentioned, we do have, active, donations coming in. People will contact us and say, hi, I have this or that I would like to donate. It's, of course, very different now. We are very, very careful about provenance. We want to make sure that whatever we take into our collection, it is, not only legal, but ethical for us to do so. So we ask a lot of questions. So one of the things that are my favorite type of object to collect are what I call primary donations. What do I mean by that? Well, if you personally have had a bit of your body removed, I will talk to you. You know, a good example for that is in the upper right-hand corner. You see that jar with that's me holding my husband's gallbladder. So he had to have his gallbladder removed, and I, of course, accompanied him to the doctor's appointment and informed the doctor that after the gallbladder was removed, I would be taking it. I didn't ask. You'll notice I just informed him that I would be taking it. So that was an interesting. But I was able to do that, and so we have that in our collection. Obviously, my husband agreed to that as well. I didn't. He was all for it. But that's wonderful for us in the sense that there's no doubt that the individual gave consent to have their body part, come into our collection because they're still alive. They sign all of the paperwork, and then I'm able to interview them and ask them specific questions about how they came to have this body part removed. What was this thing? And whatever they're comfortable revealing, we really like to gather all that information, that primary source data, if you will, because we really, really do not have that in our other specimens. The other specimens that are coming from the 19th century, the information that was obtained was really all about that specimen, not about the individual. And now we're very interested to learn about both the specimen, the condition that the person had, but also about the individual, their medical narrative, if you will. We have in the bottom left-hand corner, that is my former boss, former director of the Mütter Museum, Dr. Robert Hicks, not looking entirely happy because he's holding a jar of picked human skin. Now, this is something that I've received a lot of, teasing about over the years about why on earth would you accept a jar of picked human skin. And for the record, I haven't accepted a jar of picked human skin. I've accepted 3 jars of picked human skin. So let us be clear about that. And the reason I accepted them is because it's a very, very important educational specimen. It's very hard to use physical specimens to convey and properly illustrate a mental condition. But in this case, this picked human skin represents a condition that used to be called dermatillomania. And after the DSM Volume 5 was published, the name has changed to, and I do not pronounce it correctly, I believe, but I believe it's something like excorision disorder. This is the compulsive need for a person to pick skin off their body. And in this case, this individual approached me, and she lives in California, and it picks the skin off of her feet and saves it because she does have this condition. But she has managed to control it to the point where she only picks the calluses off of her feet. She does not cause herself any damage, but she wanted to donate the skin to us to provide an illustration, a very, very effective illustration of what this condition is. And so what often happens is people will see this jar in our collection and they'll be, oh my goodness, what is this? And then they'll lean closer, they'll look at the label, and then they're drawn into the story, into the narrative of it. One of our most recent acquisitions, one of our most recent donations was the one you'll see here, the skeleton. This is Carol Orzel. Now, Carol had a very, very rare condition called FOP, fibrodysplasia ossificans progressiva. It is extraordinarily rare. There are only about 900 diagnosed cases in the world. And Carol, before she died, made arrangements that she wanted her skeleton donated to the museum. And so I could give an entire hours-long lecture just on the process and the journey that we had to go through to make this happen. But we did. We were very happy to be able to make her final wishes come true. And we're very happy that she made that ultimate decision to donate her skeleton to us, because it is it's just an absolute joy to know that we honored her final wishes, and we were able to do it in a way that hundreds of thousands of people hopefully will be able to to see her skeleton, to learn about FOP. She was very, very active in educating about FOP during her lifetime. And that was what she wanted to be able to do after she died as well. So we were very happy to make that happen. So thank you, Carol. Now, some are borrowed. We try not to have long-term loans come in, but we have done that over the past. So the one on the left is from an institute in Philadelphia called the Wistar Institute. And of course, we do have some extraordinarily rare objects as well. So the device that you'll see to my right here is, it's called a piezoelectric device. And I believe it was the first instrument ever used to measure radioactivity. And it was actually built by Marie and Pierre Curie. And this was donated to us by Marie Curie herself when she did her grand tour of the United States. So that is a very brief overview of the College of Physicians, the Mütter Museum, and our collection. So now I'm going to shift focus and talk a little bit about what we are doing with our collection. Now, prior to the pandemic shutdown, we received on average about 188 visitors a year. Those visitors came and looked at our collection and were educated in a passive way, just by looking, which is great. We absolutely love that. It's very, very wonderful. But we're also very committed to utilizing our collection for medical scientific research. And so the rest of my talk is going to shift and discuss exactly some of the things that we're doing in that regard. So this all got started, the Mütter Research Institute, the genesis of this institute started way back in 2007 with the Cholera Project. And I wish I had a more exciting name, but that didn't occur to me to do that at the time. Again, I could talk for 45 minutes, and I have, on just the origin of this story, how it came about. But basically what happened was I got a phone call from a lovely individual in Canada who wanted to know if I had any fluid-preserved specimens of intestines of people who died of cholera. And they had apparently been calling all over the world looking for these specific types of specimens, had struck out. So they had already kind of sounded quite defeated when they had called me and said, well, we don't suppose you might have one of these. And I looked at my database and I said, I'm sorry, I don't have one of these. I have 6 of these. And I believe they dropped the phone. Hello. But once they kind of regained their composure, they were extremely excited and said, we have a proposition for you. We would like to test this this intestinal specimen. And I said, why? And they said, well, we are very interested in learning about the genetic differences of different cholera strains. So it really started me down the path of learning more about infectious diseases. And we've had 8 different pandemics of cholera that we know about. And we're currently, I believe, in our 8th pandemic of cholera. And this goes back, to the 1800s, probably before that as well. And of course, it's still a very, very serious public health concern in certain parts of the world. So it's very relevant. This isn't just a static 19th century disease that we don't have to worry about anymore. Thousands of people die a year still of cholera. The World Health Organization estimates about, potentially up to 120,000 people die a year due to cholera. And that, of course, when we say you die of cholera, we're talking about the bacterium Vibrio cholera. That is the active agent, the destructive agent for the disease of cholera. And it has many different forms. But the question is, which strain caused what pandemic? And here's the issue, is that cholera inhabits your intestines. When you die of cholera, when you die in general, one of the first parts of your body that starts to decompose is your guts, is your alimentary canal, if you will, your intestines, because you've got a living colony, your microbiome of living organisms, of bacteria and viruses and yeasts, and they're still alive. and then they'll start to just basically digest you once you die. So what happens is you lose that potential DNA very quickly, and then that is not something that you find in the archaeological record, because cholera kills you so quickly. It does not then go off into your skeletal system the way another condition, like another infectious disease like tuberculosis. We can find tuberculosis in your skeleton. Really, as of right now, as far as I know, we have not been able to isolate cholera DNA from skeletons. So we really were at the mercy of hoping that somebody had saved cholera, from a historical outbreak. And so it just so happens that we had done that, and we had specimens that dated to 1849. There was an outbreak of cholera in Philadelphia that killed over 1,000 people, and one of our fellows, had the presence of mind to actually save sections of this cholera in alcohol, and that's important to note, because they knew, obviously, they knew that cholera killed you via your intestines, via your digestive tract. But you have to remember, 1849, they didn't know exactly they didn't understand that bacteria was the reason for this. But our fellow, Dr. John Neal, said, well, I'm not exactly sure the details, but I'm going to save these sections because I want to see if I can see any gross differences in the intestinal systems, or any kind of lesions, things like that, using a microscope. And he was one of the first people in the United States to actually look at these specimens under a microscope. So what were the goals of collecting and sampling the specimen? Well, they really wanted to find out, the researchers, what specific strain of cholera was responsible for causing these early pandemics, and how will and hopefully they can get that. How will this historical strain of cholera, how will that look compared to the modern strains of cholera that are going on right now, that are still active right now? So what we did is we actually created a clean room, a sterile room, in situ at the Mütter Museum. And I remember at this point we were not set up. We did not have any scientific labs. We didn't have anything like that. We did have a lot of bleach, and we had an empty room at the time. So we actually set up, a clean room such as it was. And you can see here, there's some art in the background that was going to go up on the wall for an exhibit. We did the best we could. And then they opened up the jar, and they took small samples from that. And as I mentioned, that these were bottled prior to the invention of formalin. And let me see if I can go back. You can see here, looking at the top two specimen jars here, those were, they used a pig's bladder, a disc of lead, some parchment type paper, and some black pitch to seal the jar closed. They would put the intestine in, filled it with alcohol, and then use these types of materials to seal these jars. And why am I specific about that? Well, you just can't unscrew this jar, mess with the specimen, and screw it back on, and nobody would be the wiser. We could tell by looking at this type of sealed jar that these specimens had not been touched since they were first bottled, potted, what have you, in 1849. That's very important because, again, we knew that alcohol wasn't, formalin wasn't invented at the time. And when we were doing the DNA analysis back in 2007, it was important because any interaction with formalin could disrupt the DNA. So they were very happy that they had never been in contact with formalin or formaldehyde. So what happened? Well, we took 6 samples, or we took 12 samples from 6 specimens, very, very small. Again, you don't need a lot of tissue to do this. And what happened was 1 out of those 12 specimens actually yielded positive results. We weren't sure this was going to happen. Back in 2007, when they explained this to me, it sounded like something from Star Trek or something, again, in science fiction. But it happened. They were able to get the DNA of the cholera pathogen that was embedded in that intestine. So not only was that extraordinarily cool, but we actually got some pretty nice papers. You know, we published our results in some journals that you may recognize, the New England Journal of Medicine scientific reports, and that was what really started us on the road to creating the Mütter Research Institute. That's what really made us say, you know, I don't think this is a one-off. I think our collection has more to give to the scientific and medical community. I think we can make a difference. I think we can save lives, frankly. I think we can utilize this 19th century collection to save 21st century lives. And how are we going to do it? One jar at a time. So that's what we decided to do. And of course, once we published it, you know, other people started to contact us about different research projects, but they also contacted us because they wanted to further investigate the cholera or some other things like that. So we were approached with some other researchers who said, this is great. I should mention we were able to sequence 96% of the genome of the cholera strain, and we were able to prove what strain it was that caused the pandemic in 1849. It was the classical strain, which was the prevailing thought was that it was that, but until we did this, there was no empirical evidence to support that. It was just, we think that's what it is. Now, because of the work we did, we could say, yep, that's what it is, at least for this. But now that we knew that, that started opening up other questions, which is great. This often happens in science. You answer one question, 10 more questions get posed to you. And we love that. That's how you move forward. So the question that was presented is, okay, so now we know that it was the classical strain of cholera that caused these outbreaks in the 19th century, but we saw as we moved into the 20th century that a different strain of cholera took precedence, was dominating, and that was called the Tor strain. Well, why? What happened to this classical strain? Why did it lose dominance? Why did the Tor strain become the more prominent strain of cholera in the world? And so these researchers here had that question, and again, they wanted to look to us to find the answers. So again, we decided that it would be worthwhile to provide them with a tiny bit of tissue. And keep in mind that when we do this, we don't do it lightly. We don't just say, oh, let's crack open a wet specimen, tear off a hunk for science. No. We are very, very strict about what we call destructive analysis because we know that there's no going back. You will not get that piece of your specimen back. And so you have to make darn sure that it is worthwhile. And it's also not going to be redundant to the work that's already been done. So these researchers made a very, very good argument to the fact that now that we know the genome sequence, we need to know more specifically about the classical strain and especially to see about if it mutated in any way that rendered it less competitive against other strains. And so we decided, okay, that seems to be a good use of the specimen. And so we allowed them to move forward with that. And as of right now, there is an article that is in the process of being reviewed for publication. So I can't really talk too much about the specifics of it. I want to wait until the team presents it. All right, moving on to some other instances of research. And in many cases with research, sometimes things start accidentally. Again, that is my former boss, Robert Hicks there, looking oh so pleased. I love showing that picture of him. I wish I had more. I know I'm running short on time, so I can't give you the whole story, but long story short, never leave your director unattended. I took my staff on a field trip to another medical museum about 3 hours away in Washington, D.C. And then Robert was supposed to join us. At the last minute, he decided not to. And he took that time. He was supposed to be working on a paper. He took another employee on a tour of one of our collections areas. And it was there that they were looking through our phlebotomy collection, our bloodletting collection. And we had many, many cases there. He decided to open up one of the cases to show this employee what a lancet looks like and how they did bloodletting at the time. And what fell out? Well, a smallpox scab. So he did what any normal person would do. He put it on a paper plate and put it in his office and called me and told me to call the CDC, the Center for Disease Control. No, I've never let him forget that. So what happened was very, very quickly, we got a nice call back from some very concerned people from the CDC and the Philadelphia Department of Public Health, who said, we'll be down tomorrow to assess the situation. And they did. They were very nice. And ultimately, we ended up finding quite a few of these vaccination kits, these early vaccination kits that were just put in our bloodletting kits, because if you looked at them not very carefully, they looked like bloodletting kits. And so that was fun. So what they did is they took all of the kits down to Atlanta, Georgia, which is the capital of the CDC's, where their main headquarters is located, and kept us appraised of the situation. And you can see here, this is some of the scabs that we found. And again, if you're looking at these containers, just from the outside, they look just like a bloodletting container, but you open them up inside and you'll see that they have very specific accoutrements that are very specific to vaccination rather than just bloodletting. Things like little tin to hold the scabs, the glass plates there that you can see that would hold lymph, and of course the lancets to use for vaccination. So they gathered up all of these and sent them to Atlanta, and they kept us kind of appraised of the situation. And the CDC is wonderful. They have a top state-of-the-art DNA lab for things like that. However, their DNA lab is not an ancient DNA lab. And that's very important. It's very different. A DNA lab that's set up to do modern DNA with fresh material, let's say like a fresh blood sample or something like that, may not be adequately set up and have the tools and the knowledge and the equipment to do ancient DNA extraction, which is totally different. So the people at the CDC were very interested in knowing what type of smallpox vaccination material was used, what orthopox, what other type of pox was used to vaccinate people against smallpox. And we said, well, we actually know some people up in Canada that could do that a lot better than you could, because they have an ancient DNA lab. So we were surprised that the CDC actually allowed us to facilitate the shipment of the smallpox material from Atlanta to our partners, which is the McMaster Ancient DNA Center in Canada. They were the ones who did the work on the cholera project, so we knew to call them. They were very, very excited. And again, long story short is we were able to get the DNA of the orthopox, and we were able, we again can't give too much away, but I can tell you that our paper was just accepted for publication in a journal called Genome Biology, and hopefully it'll be coming out soon. So you'll be able to get all the details, but it was very, very exciting. And it's really inspired us to continue this research, to continue looking to our collections to find these things. And it's very important because this is what's very exciting. Not only, you saw the scab material that we had. So the CDC obliterated most of the actual biological scab material when they did their initial DNA. So there wasn't much left of it when it got sent to Canada. But I said to them, I should mention I'm a forensic scientist by training. So I was saying, from what I know about our collection, nothing was washed back in the day. Why don't we see if we can swab the lancet blades and see if there's any residual DNA? Because if we can get DNA from a alcohol preserved jar, jarred specimen with minute amount of trace DNA, maybe we can get DNA from the knife blade. Maybe we can get DNA from an empty smallpox scab container where there's no visible smallpox or no visible orthopox scabs in there. That doesn't mean they weren't in there. Doesn't mean there's not trace bits of it left. So let's use some archaeoforensics, if you will. Let's try. And since it was our collection, we decided, we kind of researched what kind of non-destructive sampling techniques can we use with the these objects, And it was very exciting because we actually got results. I mean, they weren't as good results as they would have been from like the actual scab, but we got results, which was very, very excited. And, you know, what's really comforting and good to know is that the CDC, you know, approved of this research project. They were actually, they were very, you know, encouraging of us because it helps them. The more information they have about historical strains, they can add that to their database. So it's definitely a mutually beneficial type of thing. You know, we don't want to just do cool science for the sake of it because it's cool. I mean, you know, we want it to actually ultimately lead to some knowledge that could benefit. And so that's one of the things we're very happy about. And to know now that we have the ability to do this in a non-destructive way, where we don't potentially have to obliterate a sample, very, very exciting. So again, like I mentioned, we'll be publishing this very soon in genome biology. So stay tuned for that. And another thing we're working on is kind of a cross between a historical archaeological project and modern day scientific projects. And in Philadelphia, I think it was 2016, 2017, we actually ended up rescuing a cemetery population from a developer who was putting up an apartment building right in Center City, Philadelphia. And again, I don't have time to go into the details, but long story short, you know, unfortunately in Philadelphia and in many parts of the United States, capitalism rules. And if you have a piece of land that is being developed by a private individual, so that this individual or corporation owns the land and they are using just private money to build their apartment building or their whatever, meaning there's no federal, state, or local government funding associated with this project. If they find human remains in a historical context, there is nothing that they will do to intercede on behalf of that population, of that cemetery population. So that's what we found happening in this situation, where the developer was found these human remains. They had bought a parcel of land that they knew had a cemetery in it, but that they were told, and they found historical documents to indicate that that cemetery had been moved. Well, it hadn't. You know, they just did not move the individuals. They moved the tombstones, but they didn't move the individuals. So they started digging up human remains, and they were not gentle with them. So when we got wind of this, like I said, there was nothing legal we could do about that. We couldn't call any agency to make them stop. But what we did have the power of was the media. We have a lot of friends at the Mütter Museum that are in the media, newspapers, magazines, TV, and we called all of them and we said, hey, you know, you want a story about desecration of human remains, destroying history, all these things, we got you covered. So that's basically what we did. Or I should say, we didn't do that. Somebody did it. It was an anonymous tip. So that's something so we were able to take custody and become the stewards. We did not own these individuals. We worked with something called the Orphans Court in our justice system that kind of oversees the disposition of human remains that don't have a known descendant, so to speak. So the Orphans Court placed the Mütter Museum and our consortium of institutions that took on this task as the stewards of these individuals, with the goal that we would, we excavated them. There was about 491 individuals. We're storing the remains, and we are going to rebury them in 2023. And what we were able to do is we were able to reach out to what we call the descendant community of these individuals. We knew that they were buried in the First Baptist Church of Philadelphia's cemetery, and the disposition time was between 1707 and 1858. And so we were able to actually reach out to the church that was still in operation at the time. And so we reached out to the representatives of the church and we said, we have these individuals. Here's what we would like to do. we would like to rebury them. But before we do that, we would like to do some scientific analysis of these individuals, and we would actually like to do the DNA analysis of these individuals for many reasons, to learn a little bit more about them, but also ultimately so that when we do rebury them, hopefully what we'll be able to do is we'll be able to bury entire generations of families in one specific area, so bury them together, because again, 1707 to 1858, there were multiple generations of families buried there. But so the descendant community they granted us the permission to do this type of research. And one of the things that I wanted to look at was dental calculus. And the reason I wanted to do that is that even though we were granted the permission to do destructive analysis on bone to get DNA, calculus sampling is considered non-invasive. I mean, it's destructive in the sense in that it will destroy the calculus sample, but the calculus itself is not human remains per se. It is the accretion of mineralized tartar on your teeth. But what's interesting is in that accretion is lots of DNA, lots of your DNA from like epithelial cells, saliva, things like that. So I was very interested in getting samples of this dental calculus in order to do some very specific research. And what I was very interested in is something called the microbiome. So your oral microbiome, and that is all of the bacteria, viruses, yeast, all of that, those living organisms that populate your gut, and that we're doing more and more research on, that it is very, very important in our health and our well-being and everything. It's very interesting in that about something along the lines of 9 out of 10 cells that are physically present in or on your body are actually not your own. It's your microbiome. And if you were to take away all of the microbiome, from your body, you would be 3 pounds lighter and also dead, which is very interesting. So again, just to kind of go through this quickly, what we were able to do is we were able to collect the calculus samples. We were able to process them and get the DNA of the microbiome. And it was very, very, you can see here, this is a chart that what this chart basically indicates is that we have very good microbiome samples. So we have blanks, we have controls and things like that. So we were able to say that there was a very distinct difference between our blanks and the microbiome and even the soil samples that we got as well. So it's very, very good indication that we're seeing authentic microbiome. Again, this is a very big chart. It just kind of showing the different types of taxa that are present in the samples. Very exciting because we're having lots of different variety of this, and we're very encouraged about the results. And another thing is that this whole type of research project is so exciting because we're doing something that's really cutting edge. Microbiome research is very relatively new in that, and then so the fact that we have this population, the fact that we have consent from the descendant community, and the fact that we're getting such wonderful results just from the few that we have done is very, very promising, and we hope to be able to do more. And we're also getting, like I said, excellent results. There's very little contamination. It's just all told very, very exciting. So I get very excited about this. You can see that, I'm used to wearing PPE. I wear masks all the time at work, and I got a big smile under my mask there because we are going to move forward with even more research. We can get the analysis of these things from multiple sources. We can get the oral microbiome, as I mentioned. We have the human host biome. There's even the possibilities of getting things like food sources. Again, what's embedded in the oral cavity of these individuals. We have soil samples, and from this, we're able to extract all these types of potential pathogens, infectious diseases, to kind of understand about the kind of overall health of this population. And because we have such a large sample size, over 400 individuals, almost 500 individuals, we can do something called population level analysis, where we're able to get kind of this overview picture of the health of these individuals. So again, it's very, very exciting. And so what we're going to do, the next steps that we're going to do, again, this is once we reopen, once we can kind of get our labs back up and get on track again is we're going to keep moving forward with this DNA project, and we're also going to look at the soil samples that we collected, do some comparisons. We're going to really stop and think about what questions do we want to ask. So the kind of first thing we wanted to do is, can we even get the data that we need? Can we even extract the oral microbiome from this calculus? That was the first step. Now that we know that we can, okay, so many questions. Where do we start? It gets a little overwhelming sometimes. But of course, the last point here that I always have to try and stress is that we have a lot of questions. We don't have a lot of money, probably a lot less than we even thought we did. So, of course, what goes hand in hand with science is the exploration of funding. So we're hoping that our population size, that our initial research will bring us to the attention, of potential funding institutions that will give us some much needed, money to kind of continue this very, very important research. And then we also plan to, again, reach out to other similar institutions to let them know that this is the kind of work that we're doing and this is the kind of results that we're getting. And we're very, very excited to kind of keep doing that. So one of the latest things that I just did recently, about right in late February, I was actually invited down to speak at something called PACARB, the Presidential Advisory Council on Combating Antibiotic Resistant Bacteria. They really, really like acronyms in DC. So this is an offshoot of HHS, Health and Human Services in the United States. And they had heard, apparently somebody had heard me talking to somebody else at another meeting about the microbiome research that we were doing. And so I got back to my office and I had this very odd email from this odd sounding institution that, by the way, has nothing to do with carbohydrates and bread. I was a little disappointed. And they said, we heard about your work. Would you talk in front of this this group of individuals and see, because they were very interested in knowing how can this microbiome work help inform the scientific community about something that is very, very serious, which is antimicrobial, antibiotic resistant bacteria. Right now we are very understandably directing all of our, most of our resources towards combating the coronavirus, this specific novel COVID-19. And it's very important, but there are other public health crises that have not stopped just because this one is currently going on. And one of the biggest is this antibiotic resistance. And it's estimated that in the next 10 to 20 years, the number of people that will be dying globally of antibiotic resistance, of antibiotic resistant bacteria, will exceed those that die of cancer. So again, not insignificant. And how can looking to the past, looking at our population, help inform medical researchers and doctors about what they could possibly do to prevent this? And so that's what I gave a talk on. And it went very well, and then everything shut down. So I'm hoping to be able to reach out to some of the contacts I made there and restart these talks and really, really try to convince people that these historical collections really are valuable. So this is a nice quote from Dr. Andrea McCollum, and she is our main contact at the CDC. She's in charge of the pox and rabies branch of the CDC. And she gave a nice quote to kind of really, let people know, especially scientists and researchers. Here's a funny thing I learned while talking to that is I have to use different words when I talk to the museum community about our historical specimens and when I talk to the modern scientific medical community. When I'm talking to the modern medical and scientific community, I don't call them historical medical specimens. I call them biorepository. We are a biorepository of historical pathogens just waiting to be accessed. So words matter, terms matter. I'm learning that. And that's basically my talk. Here are some of my references. Little my image disclosure. And here's a little bit of extra news. I wanted to let everybody know here that I actually wrote a children's book, and that's available for pre-sale. It's going to be available for wherever books are sold, I believe around July 28th, but you can order them through the Mütter Museum gift store right now, and I will personally sign all copies of the book that you buy through our wonderful Mütter Museum store. So that's all I have on that.